Hi students, this is A. Mandri Kumar Rajesh. In today's video, I am going to teach you two types of crosses. They are test cross and back cross. Let's start with test cross. First, I am going to give the definition for test cross. It is a cross to determine whether an individual is homozygous or heterozygous for a dominant character, and that dominant individual should be crossed with homozygous recessive parent. So, it is a cross to determine whether an individual is homozygous or heterozygous for a dominant character, and that dominant individual should be crossed with homozygous recessive parent. Why we have to do cross the dominant individual with the homozygous recessive parent? Because then only we can predict the genotype of the dominant character. Then only we can predict the genotype of the dominant character, and that cross is called test cross. Now I am going to tell you how Mendel constructed the test cross. In monohybrid cross, Mendel could observe all the young ones are tall in F1 generation, and in F2 generation he could observe both tall and dwarf plants in the ratio 3:1. So then he self-pollinated the dwarf plant from F2 generation, and he got dwarf plants in F3 and F4 generation. Then he concluded the genotype of dwarf plant was homozygous. Then he wants to predict the genotype of the tall plants from F1 and F2 generation. So he made a cross to determine the genotype of tall plants. So he crossed the tall plants from F2 generation with a homozygous recessive parent. This is how Mendel constructed the test cross. Now I am going to figure out the test cross. Here we have two possibilities of crosses. The one is capital T, and here the another one is capital T, small t. I told you earlier, here we have two possibilities of crosses. They are homozygous dominant cross with homozygous recessive parent. Then the next cross is heterozygous dominant parent cross with homozygous recessive parent. First we go with the homozygous dominant parent. So here the genotype of homozygous dominant is capital T, and here the homozygous recessive parent genotype is small t, small t, and this plant act as female and this plant act as male. Now we are going to plot the gametes in the Punnett square. So this is male, this is female. So small t, capital T, capital T, and the fusion of gametes takes place and it produces capital T, small t, tall plant. Capital T, small t produces the tall plant. Capital T, small t produces the tall plant. Capital T, small t produces the tall plant. Then the phenotypic ratio would be 4:0. 4 tall plants and 0 dwarf plants. So when a homozygous dominant plant crossed with homozygous recessive parent, the phenotypic ratio would be 4:0. 4 tall plants and 0 dwarf plants. By this, we can predict the genotype of the dominant character. If you find 4:0, the phenotypic ratio, we can predict the genotype of the dominant character. Then the genotype is capital T, capital T. Then we move into heterozygous dominant. So the genotype of the heterozygous dominant is capital T, small t, cross with small t, and this plant acts as a female and this plant acts as male, and the gametes are formed and they are plotted in the Punnett square. So this is a male, this is a female. So small t cross with capital T, small t, and the gametes are fused to form capital T, small t, tall plant, capital T, small t, tall plant, small t, dwarf, small t, dwarf, and the phenotypic ratio would be 2:2. We can simplify the phenotypic ratio into 1:1. So when a cross is made between the heterozygous dominant and homozygous recessive parent, we get the phenotypic ratio 1:1. By this ratio, we can predict the genotype of the dominant character is capital T, small t. So if we find 1:1 ratio, the genotype of the dominant character is capital T, small t. This is called the test cross. If you want to study whether the tall plants are heterozygous or homozygous, we can make use of the test cross. So this is called test cross. Now I am going to explain the next cross called back cross. Back cross may be defined as a cross performed between hybrid and one of its parent. The parent may possess the dominant character or the recessive character. Why we have to do back cross? Because the reason is during the hybridization process, the most good traits of the hybrid plant get diluted. In order to increase the good traits of the hybrid by performing back cross, so the hybrids are repeatedly undergoes back cross in order to transfer the good traits from parent to the hybrids. And there are two types of back cross. They are dominant back cross and the recessive back cross. I told you there are two types of back crosses. They are dominant back cross and the recessive back cross. First, I go with the dominant back cross. When the F1 hybrids are crossed with the dominant parent, in F2 generation we could obtain all the young ones with the dominant character, and there is no appearance of the recessive character in F2 progeny. So that cross is called the dominant backcross. Next, we move into the recessive backcross. When the F1 hybrids are crossed with the recessive homozygous recessive parent, the individuals of both the phenotypes appear in equal proportion. Here, both the phenotypes appears in equal proportion, and this cross is also called as test cross because the recessive backcross are helpful to identify the heterozygosity of the hybrids. By this information, I conclude this video session. I trust this information help you and enlighten your thoughts regarding the classical genetics. If you have any doubt, please clarify with me, and thank you very much.